Hello, my name is Rob Gilbert, and I'm the biochemistry tutor here at Magdalen College. And today we're going to be showing you what it's like being interviewed for biochemistry in Oxford. Of course, every interview is a bit different, but in general, interviews will follow the kind of pattern that this one will. Um, they will usually begin with a bit of a conversation about the interest that the candidate has in the subject of biochemistry in this case. And then building on that, we may discuss some of the things that are mentioned by the candidate, or they might have been mentioned in their UCAS personal statement about what is drawing them to apply to biochemistry. And then after that, that beginning of the conversation, based on the information really that the candidate can provide, then we move into asking some questions. But these questions are not questions that we expect people to know all the answers to. It's really about setting up a conversation. And in fact, the idea of an interview is to mimic a little bit like the experience of having a tutorial where there's material to be discussed, there's material to be agreed on, there are problems to be solved, and that process enables the candidate then to show us their interest in biochemistry and their aptitude for studying the course here at Oxford. So it's very much about enabling a student to show us what they're good at, to tell us about their enthusiasm for biochemistry, and also to draw out from them the ability to solve problems or to deal with new information that we might give them. Normally, an interview would have at least two people in it. For the purposes of our demonstration today, it's just me going to be talking to one of our current biochemistry undergraduates who will be taking on the role of the interviewee in this case. But normally there'll be at least two people in any interview. And it's worth saying that for biochemistry, every candidate gets interviewed in two different colleges. So there's two completely different sets of people assessing candidates, and that's important to the objectivity and the collective decision making of the process. So now I'd like to introduce Kian, who has very kindly agreed to be our interview subject today. Kian is a current biochemistry undergraduate, and now we're going to switch into Kian and myself talking as if we were undertaking an interview for the biochemistry course here. So, Kian, thank you very much for coming to see us today. It's great to have you, and I hope you're okay. Are you comfortable there for the interview? Is that okay?
Yeah.
Okay, fine. You got a glass of water. Very good, very good. And can I check? Have you got a piece of paper and a pen, pencil or a pen for
Yeah.
paper? Brilliant, great, brilliant. Okay, so. Now, it won't surprise you, Kian, for me to begin by just asking you to tell us a little bit about why biochemistry is a subject that you would like to study, you think, at university. You could have, applied for a number of different courses. You could have applied for medicine, biomedical sciences, chemistry. So why biochemistry? Why that course?
I think for me personally, I think the reason why I've chosen to study and apply for biochemistry is that I'm really interested in the molecular mechanisms. So I think a course like medicine was very focused on, treatments and doing actually treating patients, but I'm quite interested in, research and trying to understand mechanisms behind, a disease and cancer and understanding how our cells work. I think biochemistry specifically because I much prefer cell biology, so I much prefer looking at individual cells and how different things work inside of a cell, as opposed to looking at a multi-cell or a whole organism scale.
Yeah. Are there any particular mechanisms that you've come across that you think you've really thought, wow, that's amazing. I never thought things could be that well controlled or
I think probably from what I've been looking at, I think the most interesting would be the cell cycle, because I think it's such an intricately finely tuned process where you've got multiple different cyclins and how they are processed, where you've got one being produced and the production leading to the destruction of another. And it's sort of such a fine-tuned process where you've got so many different kinases being activated and deactivated, and then the activation of different ubiquitin ligases to degrade other ones. I think it's a really interesting process.
Okay, so tell me a little bit about that. So these kinases, so these cyclin dependent kinases, what you know, how do they kind of regulate the process? I mean, because you want to kind of link the cell cycle together with DNA replication as well, don't you? And make sure that kind of the cell doesn't divide until you've made your new copy of your DNA. So what how how is it all tied together? You know, how do those proteins interrelate? The ones you mentioned, the different because you mentioned some different proteins. You mentioned cyclins and kinases. You mentioned ubiquitinase as or ubiquitin as well. So say a little bit for me about how that all interrelates and what's going on.
Well, so during the cell cycle, at like the start of the process for cell division, you need to have some sort of like growth factor to bind to your cell receptors and to trigger the pathway, which then leads to the activation of cell division. So that's quite an important part of regulation of the process, which is that
Okay.
you need enough signal to activate downstream kinases to increase the transcription of the first like cyclin, which is CDK, which is cyclin D. Um, and then once you've got that being produced, the CDK, which is activated by cyclin B, so CDK4 or CDK6, it forms a complex with the cyclins, and then this complex is then able to phosphorylate different transcription factors. So that's, I guess, a key part there is that you have to have a growth signal to produce the cyclins in the first place. But then during cell division, I think you've got I'd say like for me, what I think the most interesting part of the regulation is to make sure that you said about the DNA replication being finished. And this is I think it's quite interesting, but I'd say what's more interesting, which I can remember better, would be the regulation of the actual mitotic chromosomes. So like during mitosis, you have to make sure they're lined up along the center of your cell before they then start dividing, because otherwise, if they're not lined up properly, you can lead to incorrect segregation and cells with more or fewer or incorrect amount of chromosomes. So I think the interesting thing about how that's regulated would be that you've got this sort of this complex which is being produced by the CDK cyclin, which is active then. So the CDK cyclin B produces a sort of complex which prevents its own inhibition. So although it activates the APC and C, which is a ubiquitin ligase, it also produces stuff which helps to deactivate it. So when the kinetochores aren't bound, this inhibitory complex binds to the APC and C and prevents it from degrading the cyclin B. So you've got this really fine tuning of until this all the kinetochores are attached, you're still producing enough of this inhibitory complex to prevent the cyclin B degradation, to really time it such that you only destroy it once the cell cycle has correctly lined up the chromosomes.
Okay. Okay. Thank you. That's a huge amount of detail. Thank you. Thank you. So now um, on your piece of paper, could you possibly draw me a molecule of benzene? You mentioned receptors. I want to come back to the question of receptors in a minute, but let's talk about a little bit of basic chemistry. You've done some chemistry at school, yeah?
Yeah.
Yeah. So would you be able to, and you've probably done benzene, you've probably done organic molecules. Yeah. Would you be able to draw me a molecule of benzene on your piece of paper?
Sorry.
Okay. Okay. No, it's okay. No, I can see it. That's fine. That's fine. That's fine. Okay. So you've put a ring in the middle. Why have you put a ring in the middle? That's interesting.
Well, so benzene in the benzene ring, you've got these three double bonds, and
Right.
I guess in the historical, like the model of like the Kekulé model, you've got individual double bonds followed by the single bonds. But nowadays, it's understood that the double bonds form like a conjugated pi system, where the
Right.
electrons are actually delocalized across the whole ring, as opposed to set into double bond, single bond, double bond, single bond. So I've drawn a ring to show that the electrons are delocalized across the
Okay.
whole structure.
Okay. They are delocalized. You're right. Yeah. And what orbitals are involved in that then? What's the orbital kind of structure that's involved, or because there are different orbitals, aren't there? That are dependent on rather that delocalization is dependent on particular orbitals. Is that right?
Yeah, the pi orbitals, is it?
Right. Okay. Okay. So these are so this is a like a uh yeah exactly. So this is a sort of a ring of electrons moving through the p orbitals, isn't it, to create the pi bonds? Yeah,
Mm.
yeah. Good. So what does that do to the overall shape of the benzene molecule? If you had to think about it in terms of like to describe its shape, how, I mean, obviously I know it's hexagonal, but I mean its overall kind of shape in 3D. How would you describe the effect on its shape?
Well, I guess it makes sure it's a flat ring, is it, as
Flat.
opposed to
Okay.
being like I guess in a normal like cyclohexane, you've got like a chair shape, I think.
Yeah. Yeah. Yeah.
Whereas
Yeah.
because of the pi orbital interactions, it has to be flat to ensure they're all
Yeah.
in the same level.
Absolutely. Yeah, that is correct. Yeah, that's right. That's right. And so what you're absolutely right. So it is flat. It isn't a chair shape, um, um, like cyclohexane. Exactly because of your pi bonding. Just before I want to kind of then talk a little bit more about this bonding, this or double bonding that we're talking about. 'Cause that's what we're talking about really here, isn't it? Or something like double bonds. But and why is it that you can't, um, those orbitals? Why is it that you can't get rotation around those orbitals? Then, if you know, what is it about the alignment of the orbitals that might mean that you can't rotate around those bonds?
Um, well, so in I can I draw something? Is that all right?
Of course, you can by all means draw it. Yeah, yeah,
Just so I'm just drawing a. If you see here, you've got
oh yeah,
this
yeah,
is my
yeah, yeah,
diagram of
yeah,
the p
yeah, yeah,
orbitals,
yeah, yeah, yeah,
and
yeah,
I guess what you've got is you've got they're above and below. So if you've got
yeah,
rotation of the bond, like a
yeah,
normal like sigma bond would, you just end up with you
yeah,
break the interaction, so the electrons are no
yeah,
longer able to bridge the two
yeah.
orbitals.
Yeah, 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 that's right. Yeah, yeah. So you've got your alignment of your bonding and antibonding orbitals. They've got and so because they're aligned, you basically it's energetically very unfavorable to rotate. Yeah, absolutely. Now I'm going to show you something actually. Now, wait there. I'm just going to show you something on your screen. All right. You should see a molecule of phosphatidylcholine on the screen. Is that right? Do you see
Hmm.
that? Okay. So out of interest, I mean, you may, I don't know whether you've come across this molecule, but have you come across phosphatidylcholine at all in what you've done at school?
Um,
Um,
I think it might have been touched on as being one of like the phospholipids in the membrane.
Precisely, yeah, that's right, absolutely. So it's one of the principal phospholipids actually forming the membrane. So now this structure, as you can say, it's the obviously the kind of chains are made of hydrocarbon sort of units, aren't they? And and you can see that it's saturated. Now, I just wanted to ask, so if I was to introduce a double bond at some point along this chain. I mean, let's say I introduce a double bond here. It doesn't really matter, but say I introduce a double bond here or here. What if I introduce a double bond? My first question is what might that do to the shape of the chain if I introduce a
Hmm.
double bond at that point?
Well, I guess linked to what we said earlier, you're not able to rotate around a double bond.
Yeah,
So I guess depending on how you when you put it in, you'd either get a trans or a cis double bond,
yeah.
which would mean that it would introduce like a kink in the chain where either it would
It
kink
it
out
would
or kink in, but
it
it
would
just sort of
it would
restricts
introduce
the
it. Yeah.
geometry.
Now, now, of course, when we learn about phospholipids and phospholipid membranes, which you'll have learned about at school, kind of with the lipids and the proteins sort of sitting in the fluid mosaic. Um, we think of those of the chains as being highly mobile now, and obviously, as you said, it's going to produce a double bond. It's going to kink it, and that will produce it'll make it a bit more rigid, won't it, as well?
Hmm. Yeah,
Okay.
I think so.
So what do you think that will do to the kind of packing between the lipid chains? What effect do you think it might have on the packing between the lipid chains to have a kink like the one that you've said, which you're right, you're absolutely right. Double bond here would introduce a kink into the chain.
Hmm.
What do you think that would do to the packing in the bilayer?
Well, I think if you've got a lipid which is more rigid and is got this kink in it, I guess it'd
Yeah,
mean that you can't pack your phospholipids as closely to one another because
yeah,
you've got
that's
this
true.
kinked
Yeah,
structure which is sort of preventing it from coming as close.
yeah, absolutely, absolutely. And final question before we move on to talk a little bit about proteins. What do you think that might do to the fluidity of the membrane? Like, you know what I mean by fluidity, sort of how
Yeah.
fluid it is. Yeah,
Well, I think introducing the double bond, if you've got less tight packing, I guess it would become more fluid because like
that's right,
the closer they
absolutely.
can
Yeah,
pack, I would assume that you'd got more like Van der Waals interactions between the phospholipids. So if you kink it, then there's less interactions, so it's more mobile.
that's great. That's great. Thank you, Keir, very much. Thank you. You mentioned when you were talking about, or you had quite a lot to say about signaling and cell cycle and so on. And at the beginning of that, you were talking about a receptor sitting in the
Mm,
membrane. And so I want to talk a little bit about proteins like that now. So like membrane embedded proteins. So membranes contain uh, lots of different kinds of proteins. Sometimes they contain receptors like the one that you were talking about, um, which kind of transmits a growth factor signal inside a cell. But they might also contain like channels and things. So my first question would be, um, thinking about what you know about amino acids and how the different characteristics amino acids have, what 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 do you think might be different about the distribution of um, of amino acids in a protein which is like the receptor you talked about, which is sitting in the membrane, which might just have like an alpha helix that it's anchored with to the membrane, and another protein which maybe is I don't know, an ion channel that signals in a neuron or in another cell. How
mm.
might the amino acid patterning differ, do you think, for those two different systems?
Well, I think for both the soluble protein and the membrane protein, you'd probably have a similar amount, like proportion of hydrophobic to the hydrophilic groups.
Hmm.
But I would assume that in the membrane protein, you've got them in a row. So I don't know how wide like how many residues would take up a membrane, but you'd have like a stretch of hydrophobic residues to make up like the helices, which
Yeah,
make up part of the protein.
yeah,
But then
okay,
also, I was going to say that if you had a channel, you'd have an inner lining of hydrophilic groups probably to
yeah,
interact with whatever's passing through.
yeah,
So that's why
yeah.
I thought the distribution would be similar, but you just have them packed in different areas.
Okay, so so by that you mean like maybe the proportion of different kinds would be similar, but the kind of their placing in the polypeptide would differ because it would the kind of environment or the role and the environment would define a need for there to be different distributions, different arrangements of them. Yeah, okay, and yeah, so that and um so, so that's right, that's good. So then if we're thinking about the folding of a protein, like
Mhm.
a folding of a protein that sits in a membrane or the folding of a protein that's in solution, then so I know this is a difficult question. I know you won't have come across this at school, so don't worry if you need a bit of time to think about it. But how might the diff how because proteins fold up, don't they, into structures, and we're going to look at a protein in a minute as a specific example. But how would the folding differ, do you think, for proteins that are going to sit in a membrane and proteins that are going to sit uh, or going to be in solution, carry out a role, like hemoglobin, say, in our blood? So that's the
Mm.
example I'm going to give you in a minute. But say like hemoglobin is in our blood, it carries oxygen, it's floating around in our red blood cells, it's soluble. And then there's other proteins that are in membrane. So how might their folding differ, do you think?
Um, well, so for proteins in our A level, we studied that for a soluble protein, you'd have I think it's called the hydrophobic effect to drive
Yeah,
the folding of
yeah,
a protein,
you would.
where you
Yeah,
have the like hydrophobic groups within your protein would sort of pack close to one another and
yeah,
form the core of a protein.
yeah.
But I guess if you think with a membrane protein, I guess it'd be more difficult because you'd probably like maybe need another protein to help insert it into the membrane or something, because
Oh, that's interesting.
I guess it wouldn't be able to just cross if you got like hydrophobic groups in the middle, but then hydrophilic groups on either side, it maybe wouldn't be able to get through without having something to help it.
That's a really interesting point, actually. Yeah, yeah. No, I hadn't actually thought you might say that, but no, you're absolutely right. That's actually a really interesting point. Yeah, indeed. You from memory, you perhaps would need a way to insert it that kind of puts it in the right orientation. Yeah, absolutely. And that gets it in, as you say, because it's going to, particularly with it being hydrophobic, it can't just find its way there in solution. Somehow it's got to be delivered, hasn't it? I guess. Yeah. It's very interesting. Now I just want to show you one more thing. I'm just going to show you another picture. This is hemoglobin. This is just a picture to sort of tell you we're going to talk about hemoglobin briefly. And
Mm.
you've probably have you studied hemoglobin a bit at school?
Yeah, yeah, we talked about the oxygen transport in the blood with hemoglobin.
Yeah, these are these heme groups here, and the heme groups they carry they as you know they carry oxygen with the help of iron. Anyway, um, this is my next slide. So um, now there's one thing I want to explain about hemoglobin. Now, if we go back to this slide, you can see that it's got four subunits. Okay, and it's
Mm.
actually got two alpha and two beta subunits, so they're colored differently here. And so you probably know that when oxygen binds, there needs to be a conformational change. The protein needs to change shape a bit in order to bind oxygen. Okay. Now it's interesting. One of the interesting things about this is that when oxygen binds to hemoglobin, and you follow it binding, you see the sort of pattern with which it binds. It follows a distinctive shape. So here we've got the distinctive shape shown here. So on the left-hand side, you can see in this graph, we've got the saturation of the hemoglobin in percentage. And on the x-axis at the bottom here, we've got the partial pressure of the oxygen. So the first question I have for you is talk about the distinctive pattern in a moment, but as you can see, there's two different kinds of hemoglobin on here, fetal hemoglobin and adult hemoglobin. Now, tell me, um, how would you say they differ? How is the adult hemoglobin different from the fetal hemoglobin?
So from the look of the graph, you've got the adult hemoglobin has been shifted to the right of the
Yeah,
fetal hemoglobin curve.
yeah.
And from if I look at the values, it looks like that it binds less tightly to oxygen. So it looks like it requires a higher partial pressure of oxygen to reach the same level of saturation.
Okay. Yeah, yeah. So that's an interesting way to put it. But yes, you're right. Yes, that's right. So it requires a higher partial pressure to kind of bind the oxygen, the adult hemoglobin compared to the fetal. Yeah, yeah, absolutely. Um, just out of interest, I mean, since I know you've told us that you're not really interested in organisms so much, more cells, that's fine. But just thinking about the system as a whole, I mean, is there a reason why you might want your fetal hemoglobin to be kind of um, bind oxygen kind of more readily than adult hemoglobin, maybe?
Mm. So I guess what I'm thinking is that obviously a fetus wouldn't be able to breathe by itself. It would rely on the blood supply from its mother
Yeah,
to provide
yeah,
to provide
yeah,
oxygen. And
yeah,
so
yeah, 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 yeah,
maybe
yeah.
it would need it need to be maybe a higher affinity to oxygen so that it could take oxygen from a mother's blood and bind it with its own hemoglobin.
Yeah, yeah, good, good. Now I want to show you one more curve, but before we get there, let's talk a little bit about the shape of the curves. Now, you've probably not done this at school, although you might have done. I mean, if you had to describe the shape of the curve, how would you describe it?
Well, I guess it looks a bit like an S, but it hasn't
Is
got
it,
like a bit in the middle.
yeah, so it looks a bit S-shaped. Yeah, so it is. Yeah, we actually call it sigmoidal curves because they're S-shaped. Yeah, yeah. Now, why do you think they might be sigmoid? And let me give you a clue. And the clue is just to think back to the fact that it's a tetramer. This thing hemoglobin. It's a tetramer
Well,
or a dimer of dimers, but yeah, a tetramer.
From what you said earlier, I think you said that the binding of oxygen causes like a change in its shape of a
Yep.
protein.
Yep.
So maybe instead of it being like a linear curve, maybe it's a sigmoidal shape because the binding of one oxygen could affect the binding of another oxygen molecule if a
Ye-
protein changes shape.
Yeah, yeah, yeah. That is true. You mean that if one subunit changes shape, it might make the other ones change shape as well.
Yeah.
Yeah, that is what people think. Yeah. Final thing then, let me show you this. So this is another curve. So I've put this in, and it's actually the sort of curve you might get in high carbon dioxide levels. So if somebody was breathing in high carbon dioxide, that's the kind of effect you might get. It's known as the Bohr effect, as you can see, as I've labeled it. So um, so first of all, to tell me, how has the curve changed? Now it's red, and then can you give me any suggestions about why it's changed like that?
Mm. So similar to the adult and the fetal hemoglobin, the Bohr effect curve is shifted to the right.
Yeah,
So I guess that from what I said earlier, that would mean that it has a lower affinity for oxygen. Is that
yeah,
right?
yeah. It does. Yeah, yeah. Why might it have a lower affinity for oxygen? Do you think this is not easy, and we don't expect you to know this. It's just we're
Yeah.
just working through these. It's an interesting problem to think through. That's all. So anything you think or suggest is absolutely valid and helpful. Why might it kind of be less good at binding oxygen now it's got the carbon dioxide in the
Um,
blood?
well, that might be, I guess, so carbon dioxide is made by respiration. So
Yeah,
maybe that if you've got a muscle or like a part of your body which is releasing lots of carbon dioxide, I guess that shows that it needs more oxygen to fuel like aerobic respiration. And so maybe the higher it releases more, so that the curve is shifted to the right so that the hemoglobin releases more oxygen where it's needed.
that's quite hadn't well. Yeah, that's sort of interesting. It's kind of thinking, kind of like almost physiologically. Yeah, yeah. But let's think a bit more molecularly.
Hmm.
So when carbon dioxide um, dissolves in water, what will you get? What does dissolving carbon dioxide do you think have? What effect does it have? Do you think, for example, on the pH of water?
Well, I remember that carbonate is one of the like carbonic acid is one of our acids
Yeah,
that we got taught about. So maybe it would
yeah,
decrease the pH. So
yeah.
there's
It
like
does decrease
release
the
more
pH.
protons.
Absolutely. Yeah, yeah. It does decrease the pH. Now, are any of the bonds in your hemoglobin likely to be pH sensitive, or are they? To think about, do any amino acids have kind of ionizable groups? I suppose is what I'm asking. Acidic groups, for example.
Yes, no, I remember that there's some which have a carboxylic acid end, so they can lose a proton or I
They
guess
can.
gain.
Yeah,
And
they
then
can.
there's some which have nitrogen, like I think lysine and histidine, so that they could
They do. They
become
do.
protonated.
Yeah, yeah. They do. They do. So if we're lowering the pH, do you think that might affect um, the well, the ionization of some of the side chains? I suppose what you're saying is yes, it might do.
Yeah, so I guess if the pH is slightly lower, it might make some of the more basic groups become protonated.
Yeah, yeah, and in fact, what people think is it kind of strengthens some of the bonding, uh, because the low pH kind of strengthens some of the bonds we call salt bridges, but those are the kind of bonds formed actually between acidic groups and basic groups in the protein, and it's thought that those get strengthened because of the lower pH. So that's thought to um, tighten the bonding. So final question. So why might thinking molecularly rather than physiologically, why might tighter bonding, as it were, holding the tetramer together, why might that result in the red curve, do you think, in high carbon dioxide levels?
Um, well, I guess what we talked earlier about the changes in the shape leading to the curve having the shape it does.
Yeah,
So obviously, I guess if you've got a tighter binding of the tetramers, it might perhaps make it slightly more rigid and
yeah.
affect if it maybe like prevent or make it harder to change the shape of the protein.
Thank you. Thanks, Kian. Thank you so much for your time today. And I hope, yeah, I hope you have a good rest of your day. Thanks, Kian.
Thank you.
Thanks.
Nice to meet you.
Nice to meet you.
Bye.
Bye. So we should talk a little bit about how that went. And maybe it'd be very interesting to know. Obviously, want to know what you thought, Kian. What I would say is when we got into um, the sort of problem solving, benzene and all that onwards to the end, it was very like actually what you might find in an interview for an undergraduate place. Um, the kinds of problems which were about taking things that people have learnt at school, like benzene at school. People know about the lipid mosaic model for membranes from school. They know about lipids from school. So taking stuff that we know from school and then just thinking a little bit more with it and putting it together with other ideas, that was very like what you might do in an actual undergraduate interview. The first part was quite like it in the sense that we started from something that Kian had perhaps mentioned on his personal statement, or he told us he volunteered about being interested in the cell cycle. Of course, Kian knows more about the cell cycle than the people generally would at school. Uh, so in some sense, there was a lot more detail in that section than you might expect in an interview. That said, two things. Firstly, sometimes candidates do know a lot about something or other they've read about, and that's absolutely fine. But secondly, it's not so much the level of detail that matters to us. In fact, it's not really the level of detail, it's more the interest that it's shown. So if somebody kind of happens to know a lot about something that they've read about, and it could be anything, there are different things people might read about, they might know a lot about it. That's good. But the key interest for us is it shows interest. It
Mm.
shows that somebody is engaged in the topic, engaged in the subject, and really wants to study it in detail. It's not about knowing lots and lots and lots of stuff. It's about the fact that the reason they know this stuff shows there's an enthusiasm there. So you don't need to know a whole load of detail. You just need to tell us why it is you're interested in biochemistry, I would say. And it's not about kind of more and more information kind of makes a stronger and stronger case for being offered a place. That's not how it works. It's more, how do you handle problems? How do you think through things? Are you interested in the subject? Can you think on your feet? When we slightly change the direction of the conversation, can you pick up on the sort of how the direction's gone and why we've moved from, say, talking about membranes to talking about membrane proteins and why they're different? And can you make sense of the links? Which is all stuff which is based on core information that is not very detailed, but helps us to assess someone's aptitude and potential for studying at Oxford. That's what I would say. I don't know what you think, Ian.
Yeah, no, I definitely agree that I think the most important part of showing at the start of the interview is just to show that you really care about the subject. And I remember for my interview, I don't think I knew very much specific biochemistry in a lot of detail,
No.
but it's more that I remember from my St Hilda's interview at least that they asked me a couple of questions about epigenetics because I talked about that in my personal statement. And even though I feel like now I know a lot more about actually how epigenetics works and we studied it a lot more, but I think it was just more that I could show that I was interested and I'd read this book
Yeah,
about it and this is why that's part of the reason why I'm really interested in biochemistry.
absolutely. Absolutely. Yeah. Um, yeah. And the other thing I'd say is that I think quite often you'll find that in an interview, candidates are shown something in the interview. So I showed a molecule of a lipid and I showed hemoglobin and those binding curves. And it'll vary. Sometimes it's an image of a cell, sometimes the questions are a bit more genetic, but it's all around. So what I mean is talk about DNA or how genes are organized. But the point is that it's not about what you know, it's about taking information that you already have come across at school and developing it, and then giving you new information that you might not have thought about in order to develop that, take it forward, and see what you make of it.